So I'm going to be talking about random polypeptide sequences and their relevance for the origins of life. And I'm just going to start straight there without not much introduction. So I would like to ask you to imagine first that the circle here includes all the biological sequences that our life uses. And we know that these sequences occupy only a very tiny part of the possible sequence space, and of course this is terribly out of the range, but this is basically just to illustrate what we are interested in. So we are interested in what actually lies outside this box or this circle, at this case, in the random sequence space, and especially when it comes to what could have kind of been lying around during the early origins. So to start with our exploration, we basically looked at, we performed kind of like a scarce experimental sampling, looking around the biological space at some specific sequences that are out there. And to start, we generated 10,000 random sequences. They were all of the same length, 100 amino acids, and the natural amino acid distribution. And we performed secondary structure prediction on all of these using multiple predictors. And based on these predictions, we sorted the data out and selected some sequences that we looked at experimentally. And these results show the bioinformatic prediction of the secondary structure. And the first panel is our random sequence dataset, and we used 5 different predictors and looked always at the alpha, helical, and beta sheet content. And we did the same thing for some control groups. The first were fragments from the PDB database, then fragments from the UniProt database, and also fragments from the database of disordered proteins. And the short message here is that secondary structure actually seems to be quite abundant in random sequence space. This plot here shows the same random data set in a slightly different way. So on the y-axis here we have secondary structure content, and on the x-axis we have the predicted disorder of these sequences. So basically we see a whole range of sequences in our random data set, and we have some sequences with high secondary structure content and some with low secondary structure content. And we selected 45 sequences here. Those are the ones in color for some experiments. In short, this plot here summarizes all these experiments. So we looked at expression and solubility in E. coli and also secondary structure using CD on purified proteins. And I don't really want to go into much detail. There is a lot of detail there, but to summarize, we basically confirmed that secondary structure is quite abundant in random sequences, and we did not really expect to see that. And also, the random sequences that actually have high disordered content seem to be better tolerated by living cells. They have low aggregation propensity, and we hypothesized that these could actually make them better progenitors of soluble and functional proteins. So basically, based on this little search, we concluded that it's worth looking out there. And in particular, we decided we're going to do this in a more systematic way. We are interested in what we could find from different alphabets, and especially those amino acid alphabets that could be more relevant during early origins, during evolution of proteins. So basically, we wanted to explore different subsets of the protein, of the sequence space. So our libraries are designed on a DNA level using degenerate codons. We started collaborating here with Klara Hlouchova from ELSI. And the degenerate codons basically control the amino acid composition of our libraries. So we can design libraries from different subsets of amino acids. And to do this as precisely as possible, we developed an algorithm for this called the COOT, the Degenerate Codon Optimization Tool. And this is roughly what it looks like. So what we can do is basically find solutions to almost any alphabet and library length. We can also optimize the codon usage for expression at different organisms, and also remove some codons for reassignment to be able to bring in some of the unnatural amino acids. So amino acids that could be prebiologically very relevant but are not part of our genetic coding system at the moment. So using this tool, we basically started making some of these libraries. And I guess one of the newest thing here is that we have been able to express enough quantity of these libraries using cell-free expression system and purify. This is a sample of one of these libraries, and I know this looks very ugly, but the reason is that we have a whole distribution of molecular weights within this band. So that's basically what you see here on the multispectrum as what we have here in the sample. And of course, we are interested in libraries that would best mimic what was around, kind of lying around during the early origins. And while we do have some ideas what libraries we would like to make here, I also wanted to use the chance of actually being here, being able to present, and thank you to the organizers for that, and kind of invite you to give us some feedback on this, because we are quite often asked, like, why these amino acids and why not the others, and why this long and not that long. So I would like to really invite you to come and talk to us at some point and tell us if you have ideas about what is more relevant. And before I do that, I'd like to acknowledge especially my coworkers, Klara Hlouchova from ELSI, and also Stephen Fried, who is newly working with us on this project from Johns Hopkins University. I would also acknowledge the people that I work with, especially these guys from my group at Charles University, that's in Prague. And it's an ancient university, but we work at this new campus close to Prague. So thank you for your attention. Thank you for your attention.
Thank you, Klara. We have time for a few questions. Sure, go ahead.
On your first couple slides, there you had some data that had secondary structure on the y-axis and protein, something on the x-axis.
Mm-hmm.
Were there any constraints in that data set about the length of the peptide? Like how big were those sequences? Because to me it seemed like if you had a long sequence, it's inevitably going to pull.
Mm-hmm.
Was there any constraint on that?
Yeah, they were all 100 amino acid long. Mm-hmm.
Go to the side.
So it's interesting you found so much structure since we assume they're intrinsically disordered. I wonder how long it took. I picture peptides being a lot shorter in the early years. So I wonder how far along it would take to get to 100.
Yeah, so the first study that I talked about was basically just looking at what we can find at the random sequence place, but not that much relevance to origins of life. So we chose that line of protein basically just because it's easier to work with, and it was for us the first kind of little search. So the proteins that we want to be looking at now would be much shorter, ranging from something between 20 and 60 is what we imagine.
Toward, on the aisle and then towards the back. Yeah, sir.
Yeah, okay. So just a comment on the beginnings of this, not necessarily a question. Keith Dunker has done a lot of work on trying to figure out the disorder proteins in the world, and he has come to the conclusion that actually this disorder that we observe now, and that I presume is being predicted now by these tools, is actually design disorder. It's not disorder. Right? So it's not random disorder. So I'm not that surprised about secondary structure, but the fact that you get a significant level of disorder in your random space, that I find very strange.
Yes, thank you. This is something we have been thinking about a lot and talking to the disordered kind of protein community about it. And I do agree that the disorder that we see is different than the disorder that they see. First thing, there is a very strong compositional bias in today's IDP proteins that are mostly eukaryotic. And of course, it's usually very functional. What we see is a different case of disorder.
Down the back. Yep.
Hi, I'm Tony Beaton from Minnesota. Tell us about the field.
Hi, Matilda. Yes, so we have so far been looking at comparing the full alphabet with different versions of what's considered the early alphabet, meaning mostly just those amino acids that are biological today coded and were considered to be prebiotically available. So that's what we've been doing so far. But it's mostly work in progress. Yeah.
So I had a question. So for your newer libraries, do you have a way of dealing with sort of single nucleotide insertions or deletions? Because I could imagine that if your library, say when it's transcribed in your cell-free translation system, picks up a single insertion or deletion, it throws off your whole codon design. Is there something about the system that would prevent those from coming up?
Yes, that's a good question. It doesn't seem to be happening too much within our new library, so we then use a lot of PCR, not too much anyway, and try to use high fidelity enzymes on the way. But after sequencing our library on the RNA level, we don't see a lot of that happening. In fact, almost nothing.